Hello everyone. This is a short tutorial on PHASTER. PHASTER stands for Phage Search Tool Enhanced Release and is a significant upgrade to the popular FAST web server, which provides rapid identification and annotation of prophage sequences within bacterial plasmids and genomes. Bacteriophages are composed of proteins that encapsulate a DNA or RNA genome and can have relatively simple or elaborate structures. Phages replicate within their bacterial host after injecting their genome into its cytoplasm. A prophage is a bacteriophage genome that's been inserted and integrated into the circular bacterial DNA. This is a latent form of a phage in which the viral genes are present in the bacterium without causing disruption of the bacterial cell. PHASTER is able to identify and annotate these prophage sequences within the bacterial genomes and plasmids. PHASTER is much faster, more efficient, and user-friendly than FAST. In fact, it's more than 4 times faster than the previous version when analyzing a bacterial genome. For example, it can now process a typical bacterial genome in only 3 minutes from the raw sequence alone, or in a 1.5 minute when given a pre-annotated GenBank file. This is because of a number of hardware and software optimizations. For example, the addition of more than 120 CPUs to the FASTER compute cluster, automated algorithms to reduce both size and redundancy of the FASTER database, and the ability to perform automated lookups against more than 14,000 previously FAST and FASTER annotated bacterial genomes. All of these combined have made the backend of FASTER almost 3 times faster than the previous version. FASTER's web interface has also been entirely rewritten, making it much more visually appealing and user-friendly than the previous version. Let's take a look at this new interface. To navigate to the FASTER website, simply open up your favorite browser and type in faster.ca. This will take you to the FASTER homepage. The ribbon at the top of the FASTER webpage contains a number of options to navigate the web server. A couple of things to note here is that the help page contains a step-by-step tutorial on how to use the web server, and the About tab contains information on input and output examples. The My Searches tab can also be used to view saved searches that you might have completed in the past using this browser. Now that we know how to navigate through FASTER, let's go through the submission process. Click on the New Search tab to start a new search. FASTER has 3 input options. You can upload a GenBank formatted file or nucleotide sequence file in a FASTER format. Enter a GenBank accession or GenInfo number, or paste in a raw or FASTA formatted nucleotide sequence. For the purposes of this tutorial, let's try running an example. The results for your submission will be displayed like this. The header at the top of the page contains information about your submission, and you can download the complete set of results in a zipped folder or view them in these 3 files: a summary file, a detailed file, and an interactive genome viewer file through which the results can be analyzed. The summary file contains a color-coded table based on scores for each region. The green rows indicate scores above 90 and are categorized as intact. Blue rows contain regions which scored between 70 and 90 and are called questionable. Red rows are categorized as incomplete with scores below 70. The methods which were used to calculate the scores can be found at the bottom of the page. The table itself is organized by region number, with the region length and completeness score right beside it. The total number of proteins is listed besides that, and the region position refers to the position of a particular region within the submission file. Clicking on this will open a pop-up containing the full sequence for that region. The name of the most common phage matched with each region is listed. Next to that is the percentage GC content for that region. Lastly, clicking on the details will open up a window containing the details for that region. The column headings along with their meanings are listed at the bottom of the page. The region details contain all the information in the table, as well as more specific information for that region. Note that the exact completeness score for the region is listed here. Let's move on to the detailed file. You can download the results of the detailed file as a text file or view them here in the browser. The color code here refers to the database that the CDS positions align to. Purple is the bacterial database or GenBank file, and pink denotes the prophage database. This table is organized by the number of coding sequences within each region. Each region has its own table here. The CDS position is shown here as well as the specific blast hit. The E value is also shown to indicate the reliability of the blast hit. Clicking on the sequence will open up a pop-up with the full sequence in the CDS region. Now let's take a look at the genome viewer. The results of the submission are shown here with the regions highlighted according to the scores in the summary file. Options for the details shown in the genome can be selected here, and the file can be downloaded as an image. Clicking on a region will show details about that region. Clicking on this region populates this square with the details about this region. It will also open up a linear view of that region below. Here, the specific loci within the region are annotated for analysis. A Viewer Options table exists here as well. The arrows at the top of this table allow you to scroll through the regions. This image can also be downloaded. There is also a legend describing the type of loci within the image below. Clicking on a locus will populate this box on the left-hand side with details on that locus. This data can also be downloaded as a text file. This submission process is useful for submissions of individual files or a few sequences. However, if you have lots of sequences to run, the FASTER URL API can be a powerful tool. This URL API can be easily accessed or integrated into a user's local program. You can use any HTML request tool for Windows, Linux, or Mac. You can post to faster.ca by uploading a FASTA DNA sequence. You can also post to faster.ca with a multi-context assembly file. To do this, make sure to set the context option to 1. The GET function of the FASTER API allows you to get the status or the results of your job. If a job ID for a submission is given, the status for that submission will be returned, and if the job is complete, it will return the results. The full instructions on how to use the URL API for FASTER can be found under the Help page. This has been FASTER in a Glance. Thank you for watching.